Hi, welcome all. Today I am going to discuss about isolation and screening of industrially important microorganisms. We will talk about industrially important microorganisms, its isolation and screening. Here some of the industrially important microorganisms and their products are given. Streptomyces is an important microorganism in the industry. It is used for the production of cyanocobalamin, tetracycline, streptomycin, and neomycin. Cyanocobalamin is a vitamin, and the others, tetracycline, streptomycin, and neomycins, are antibiotics. Second example, Bacillus subtilis. It is used for the production of alpha amylase. Alpha amylase also produced by Aspergillus niger. It can produce citric acid also. Another important microorganism is Clostridium acetobutylicum. It is used for the acetone butanol fermentation. The next one, Saccharomyces cerevisiae. It is used for the industrial production of variety of products like bread, wine, beer, SCP, etc. Then chlorella and spirulina used for the SCP production. Acetobacter aceti can produce acetic acid. Nocardia used for the production of rifamycin. Then Aspergillus oryzae used for the production of amylase, cellulase and SCP. Then another one, Penicillium chrysogenum used for the production of penicillin. It is an antibiotic. Then Rhizobus can be used for the production of steroids. Gibberella can be used for the production of gibberellin. Bacillus subtilis for bacitracin. And finally Clostridium for acetic acid. Isolation of industrially important microorganisms. The microorganisms of industrially importance are generally bacteria, actinomycetes, fungi, and algae. These organisms occur virtually everywhere. Example, in air, water, and soil, and on the surface of plants and animals. But the good source for the isolation of microorganisms are soils, lakes, and river muds. 1 gram of soil contains 10 raised to 6 to 10 raised to 8 bacteria, 10 raised to 4 to 10 raised to 6 actinomycete spores, and 10 raised to 2 to 10 raised to 4 fungal spores. A variety of complex isolation procedures have been developed, but no single method can reveal all microorganisms present in a sample. Many different microorganisms can be isolated by using specialized enrichment culture technique. The enrichment culture techniques are designed for selective multiplication of only some of the microorganisms present in a sample. The common techniques used for the isolation of microorganisms are direct sponge of the soil, soil dilution, gradient plate method, that is pour plate method and streak plate method, then aerosol dilution, flotation, and centrifugation. You are familiar with the general method for the isolation of microorganism from the soil or water that is given here. First step, the sample that is diluted with sterile water. Okay, the sample may be soil or water that is diluted with sterile water. Then an emulsifying agent tween is added. The sample is thoroughly mixed and then allowed to stand at room temperature. The supernatant is diluted, that is 10 raised to minus 1 to 10 raised to minus 10. Various culture media are inoculated with diluted samples and then incubated. So the colonies form. Colonies from the plates are isolated and identified that colony. This colony maintained and preserved. Okay, this is the general scheme for the isolation of microorganism from the soil or water. Okay. Next, let's move on to screening techniques. The detection and isolation of high yielding species from a natural source material, example soil, is called screening. Okay. The isolation and detection of high yielding strain from a natural source is called screening. There are two types of screening methods: primary screening and secondary screening. Okay. First screening method is primary screening. Primary screening consists of some tests required to detect and to isolate a new microbial species that exhibit the desired property. Okay. It is needed in case of certain microbial species. Example, the microorganism that is capable of producing organic acids, antibiotics, vitamins, amines, etc. Okay, so the primary screening involves some test. The test is carried out to detect a special microorganism that have the special property. Okay. For different techniques or tests are involved. First one, crowded plate technique. Second one, Auxinography. Third one, enrichment culture technique. And finally, use of an indicator dye. First screening technique, crowded plate technique. It is the simplest screening technique and it is used to isolate and detect antibiotic producers. Antibiotic producers from soil or other natural source. The procedure involves series of steps. First step, serial dilution of the soil or other material for antibiotic producers. Second step, spreading of dilution on nutrient agar plate. The agar plate that shows 300 to 400 colonies per plate are selected for isolation of antibiotic producers. The antibiotic producer can be indicated by the presence of a zone of inhibition around that colony. The colony that shows zone of inhibition is subcultured on a nutrient agar medium and that colony purified by streaking method. This method also requires further testing to confirm the antibiotic activity associated with the microorganism. It is because of that the zone of inhibition may be due to other causes. This simple screening technique is improved by use of a test organism. The test organism may be Staphylococcus or other organism. Okay. Procedure. First step, serial dilution of the soil. Then spreading of the dilution on a nutrient agar plate. Then the the agar plate that shows 300 to 400 colonies per plate are selected. Then that agar plate with colonies after incubation are flooded with suspension of test organism. Okay. Agar plate with 100 to 300 colony after incubation are flooded with the suspension of test organism. It may be Staphylococcus or other organism. Then again incubate that plate. The formation of inhibitory zone around certain colonies indicate antibiotic activity. Then the diameter is measured in millimeter. That colony isolated and purified. Okay. Second method is, or the second primary screening method is Auxinography. This is used to detect microorganisms' ability to produce the growth factors. That is, the microbial ability to produce amino acids or vitamins. There are two major steps. Step 1, preparation of the first plate. Then step 2, preparation of the second plate. Okay. First, preparation of the first plate. Okay. A filter paper strip with 1.5 into 12 centimeter. Okay. Is put across the bottom of the petri dish in such a way that the two ends pass over the edge of the dish. Okay. A filter paper disc of petri dish size is placed over the paper strip on the bottom of the dish. Okay. First we placed a filter paper strip. Okay. Paper strip. Then above that a filter paper disc of petri dish size is placed. Okay. Then nutrient agar at 45 degrees Celsius is poured on paper disc in dish and then allowed to solidify. Serially diluted sample is spreaded on nutrient agar plate and incubated. It produces isolated colonies. This is the preparation of the first plate. Okay. Once more, first a petri dish is taken, then above that a filter paper strip is placed across the bottom of the petri dish and the two ends of the paper strip are passed over the edge of the dish. Okay. Above that a filter paper disc with petri dish size is placed over the paper strip. Okay. Then nutrient agar is poured. Then serially diluted sample is spreaded. Then incubated that plate that produces isolated colonies. Okay. This is the preparation of the first plate. Next, step 2, preparation of the second plate. Okay. For the preparation of the second plate, a minimal medium lacking the growth factor is seeded with test organism. Okay. That is, test organism is inoculated into a minimal medium. That minimal medium lacks the growth factor, amino acid or vitamin. Okay. That seeded medium is poured on the surface of a petri dish. Then the plate is allowed to set. Okay. Then the agar plate prepared in step 1 is lifted out with a spatula and placed without inverting on the surface of the second plate. Okay, second plate prepared in step 2. Okay, so the first plate prepared in step 1 is placed over the surface of the second plate without inverting. Okay, so the growth factor produced by colonies present on the surface of the first layer of the agar can diffuse into the lower layer of the agar containing test organism. Okay, the zone of stimulated growth of the test organism around the colonies is an indication that they produces the growth factor. This is the Auxinography method. Okay. So the growth factor produced by the colonies present in the agar plate that is prepared in the first step. Okay. That growth factor diffuse into the second plate. Okay. Second plate that contains the test organism. The zone of stimulated growth of the organism around the colony forms. Okay, that zone indicate the production of growth factor. Okay, let's move on to enrichment culture technique. It is a primary screening method and it is designed by Bejernik. Enrichment culture technique used to isolate the desired microorganism from heterogeneous soil microorganism. In this method, either the media or the incubation are adjusted in favor of the desired microorganism. So the growth of the unwanted microorganism can be eliminated. Procedure is simple. First step, a nutrient broth with an unusual substrate, example cellulose powder or other chemical, are inoculated with the microbial source material. Microbial source material is soil or other source material. Okay. Then that nutrient broth is incubated with that source material. Then a small portion of the inoculum from that flask is plated into a medium having the same composition, that is nutrient agar with cellulose powder. Okay, so the colonies appear after the incubation. That colony is subcultured and further tested. Another example for the enrichment culture technique is the screening of protease enzyme producers. Procedure is simple. First step, serial dilution of the soil. Then all serial dilutions are heated at 80 degrees Celsius for 10 minutes. Okay, 80 degrees Celsius for 10 minutes. At that temperature, all vegetative cells will be killed, but the spores are unaffected. Then 0.1 ml of the sample spreaded on nutrient agar that contains the protein casein. The medium pH is 10 to 12. Okay. So the media contains casein protein. Then incubate the plate. After the incubation, a clear zone formed around the colony are subcultured. That colony have the ability to produce the protease enzyme. Protease have the ability to degrade the protein or hydrolyze the protein. Okay, here the protein is casein. That casein is provided in the media. Okay, so the protein is hydrolyzed by the protease enzyme. That protease enzyme is produced by the organism present in the soil sample. Okay, so a microorganism with the ability to produce protease enzyme is isolated. Then that colony can be subcultured, purified and stored. Okay. Fourth method is use of an indicator dye. Okay. pH indicating dyes are used for detecting the microorganism that is capable of producing organic acid or amines. Okay. Example for the pH indicating dyes are neutral red, bromothymol blue, etc. The pH indicating dye's color change according to its pH. Okay. So any deviation from the pH causes a color change in the dye. Okay. Procedure symbol, that is the dye is added into the nutrient agar media, then the inoculum is added to that media, then incubate that media. So the color change in the dye occurs. That indicate the cells produces the organic acid or amines. Okay, so it is a simple procedure. The cells that produces organic acid or amine are subcultured. Then that colony requires further testing. Okay, it is not a conformational test. It is a primary screening or the primary method to detect the production of organic acid or amines. So the primary screening methods are over. Next, let's move on to secondary screening. It is a systematic screening program, and the secondary screening method used to isolate industrially useful microorganisms. That is, primary screening does not provide much information, and the secondary screening method helps to detect useful microorganism in the fermentation. Okay. The secondary screening method helps to sort out microorganisms that have the real commercial value, that have the real applicability in the fermentation. Okay. And it provides information about the product produced by the microorganism is a new on or not. And also it gives an idea about economic position of the fermentation, effect on different components in a medium, and the optimal conditions for the microorganism, and the genetic instability of the microbial culture, and the number of products produced in a single fermentation. Okay. Secondary screening helps to give information about the solubility of a product in different organic solvent, and also it gives information about the physical, chemical, biological property of the product. Okay. And also it helps to determine the structure of the product, whether it is a simple, complex or macromolecular structure. Secondary screening gives answers to many questions, but primary screening does not provide much information. Clear? So the secondary screening method is more relevant, more informative, and more applicability. Okay. Next, we can discuss secondary screening on agar media. Okay. An example is given here. Streptomyces screening, or the method is called giant colony technique. Streptomyces have the ability to produce antibiotic. That is, streptomycin isolate is streaked in a narrow band across the nutrient agar plate. Okay. Then that plate is incubated so that streptomycin growth occurs. Then after the incubation, the test organism is streaked from the edges of the plate up to but not touching the streptomycin growth. Then again incubate that plate. So the inhibitory zone for each test organism is measured in millimeter. Okay. From this experiment, we can isolate streptomycin with antibiotic producing capacity. Another method is paper disc agar diffusion assay. It is used to determine the amount of antibiotic present in a sample. First, the streptomycin inoculated to liquid media. Then the media is incubated at room temperature. The flask is aerated by mechanical shaker. So the growth of the streptomycin and antibiotic production occurs in that media. There are some tests are required to carry out, that are checking for contamination, checking the pH, estimation of nutrients, and assay of antibiotic. The results are pointed out to determine which medium is best for the antibiotic production. Okay, so these are the methods used for the secondary screening. Okay, I hope you got an idea about industrially important microorganism, isolation of the industrially important microorganism, and its screening methods, primary and secondary screening methods. Okay, thank you for watching this video.